Say hi, good morning, everyone. So I will be talking about studying mechanism and developing treatment for GRIN to be autism and using iPSC models. So the patients that we're working with, we have a total of 5 patients. We can see here, their mutations. So different types of mutations, it's 5 different mutations. Including a missense mutation, several nonsense mutations, a splice variant, and an inversion mutation. And the patients have different disease phenotypes, starting from mild to moderate intellectual disability to severe intellectual disability. And we can see here the effect of the mutation. So the method that we're using in the lab are disease modeling, are patient-derived neurons. So Professor Shani 2012, the Nobel Prize in Physiology or Medicine for finding that we can reprogram adult cells into induced pluripotent stem cells. It's like taking the cells back in time after they have already differentiated. They're now stem cells. And we usually start either with a blood draw or a skin puncture. And we make, so here these are the PBMCs from the blood. We isolate the PBMCs, these are the white blood cells. And then we reprogram them using the Yamanaka factor into induced pluripotent stem cells. And then we can either differentiate them into monolayers, neuronal cultures, or 3D brain organoids. And the advantage of this model, it's a precise disease model in the sense that the patient have the neurons in the end have the exact same DNA as the patient. So then we don't have to develop a specific mouse model for each of the mutations. And also sometimes, in psychiatric disorders, especially, there is no one relevant mouse model because sometimes there are many genes that are affected. So here we can see how it looks. So we start from the iPSCs. Here they're stained for pluripotency markers. So this is many cells. These are thousands of cells. And then we make the embryo bodies, which are 3D kind of floating spheres. And then we can either embed them into matrigel, and then they become organoids, or we can dissociate them into neural progenitor cells. So here we can see the progenitor stained for progenitor markers. And then we can further differentiate them into neuronal cultures. So in the method that we use for this study are a patch clamp, wholesale patch clamp, and calcium imaging. And this is a really large body of work. We're talking about approximately 6,000 patch cells, and I will show because of all the drug screens, and approximately 50,000 minutes of recorded and analyzed calcium signaling. So this is just generally wholesale patch clamp. It was invented in the late 70s by Ner and Ekman, and Sackman, who received the Nobel Prize for it. And it allows to record specific currents, even specific ionic channels. And it currently is the only way to measure specific currents from a single neurons. And we can see, this is as Jan showed, this is the circuit. So we have the cell, we have the pipette with the electrode inside. Once we go near the cell, we apply a small suction, we fuse with the membrane and with the cytoplasm, and then we can measure specific currents and voltages from each cell. This is the amplifiers and the analog to digitizer, so we can record them in the computer. And then we can measure, for example, the action potentials. This is the way the neurons communicate with each other. Or we can measure the currents. So once we voltage clamp them, we can measure the currents, and we can see here downward deflections are sodium currents, upward deflections are potassium currents. So this is how, for example, calcium imaging looks like. So we incubate the cells with calcium-sensitive dyes. And then we can see the calcium entering the cell, which is the activity of the cell, as changes in the fluorescence. So we can see here, for example, and I will talk about it more, untreated patient neurons, and also this is specifically Elsarin treated patient neurons. And we can see also in the raster plots, these red lines represent network bursts where most of the neurons fire together. And here each line is like one neuron that is analyzed, and all these black dots are the activities. So we can further analyze the calcium transients looking for network properties. So for example, here, this is the imaging that we're doing with them in the microscope, but then we can analyze how the network, according to the correlations between the neurons. So this is, for example, a control network, and this is, for example, one of the patients, the 7843 neuronal network. We can see it's much less connected, it's more sparse. But once it's treated, it becomes more similar to the control. And also other things that we can do with this, we can see how information propagates over the network. So we can see, for example, in untreated, the information propagates much slower than treated culture. So this study, we used a very large drug screen. So we used approximately 20 drugs. And in addition, we did several drugs together, or 2 drugs together. We worked this list of drugs. We were trying to look for NMDA modulators, general neurotransmitter modulators. We looked for drugs that reduce neuroinflammation, other drugs that were recently reported to be effective in autism. And so you can see here, we have 3 of these pages. Okay, we can see, some of these, the L-serine, D-serine, some of them are not, or pregnenolone sulfate. Some additional drugs, we have, you can see the CBD oils, lion manes, this is a mushroom, and others. Folinic acid, which has gotten some attention recently, soticlestat. And these are drugs that we also tested that they're usually a bit harder to get to the lab. And we're also doing some combination treatments. And I will show some of the results. So we start when we differentiate the cells. It takes a really long period to get the cells to mature. One of the things that we notice, and this is something that we repeatedly see in autism patient neurons, is that we have an early phenotype and then we have a later phenotype. The early phenotype are when the neurons are immature, when they're approximately 4 weeks of age, and I will show this phenotype. And then we are also looking at them later when they're more mature, when approximately 10 weeks of age. So when they're approximately 4 weeks of age, if we look at what we are doing with the patch clamp, this is an example with minocycline. Okay. So if we look at the control, so here we're measuring the excitability of the neuron, which means we current clamp the cell, we measure the evoked action potentials, these are the spikes, and then we just count the amount of spikes. And we can see when we look at this is the inversion mutation, patient with the inversion mutation, and we can see when we look at the neurons that are untreated compared to the control, they're hyperexcitable. And we have different types of measures of the excitability. Here we measure the maximum number of spikes, here we measure the total spikes. And for example, with minocycline, this reverses this phenotype. So we can see here, these are example recordings, the control, the untreated, and the minocycline. And we also measure excitatory postsynaptic currents. This is a measure of the synaptic activity, or how connected are the neurons. So we can see here, again, this is example, the control, and untreated. So all these downward deflections are the synaptic currents entering the cell. We're patching the cell at -60 millivolt, which is approximately the resting membrane potential. This prevents the neurons itself from spiking. And then we can measure, these inputs currents going into the cell. And we can see with minocycline, again, this is an example, minocycline reverses many of the phenotypes. And this is just an explanation about the synaptic activity. We can see these currents are the inputs from other cells. And now that we've done many types of these drugs, and these are all summarized together. So we can see here the control, okay? This is the untreated patient. This is still the inversion mutation. And then all these treatments that some rescue the phenotype, some rescue partially the phenotype. Importantly, because these are many tests, of course, we did multiple hypothesis corrections. And we can see, example recording. So this is the 2 types of excitability, total evoked action potentials and maximum number of spikes. And we have the control, untreated, and luteolin, for example. This is an example. EPSCs, again, we look at the frequency, how many of these in a certain time point, in, for example, 1 second, and also the amplitude, how big are these lines. And again, we can see, all these drugs that either partially or fully rescue this specific phenotype. What about calcium imaging? So here we have different measures that we're looking at. For example, we can look at the network bursts, which are the network bursts are when the neurons fire together. So we can see that we mark them with these red lines. And this is an example of a control, untreated, and this is minocycline plus CBD. And we can see, again, here we're measuring the frequency of the network bursts. Here we're measuring the size or how many neurons are participating in this network burst. And other features that we can look at are, for example, the calcium events frequency, not the network, but each of the cells, and the amplitude of the events, and more. Okay, so we have different features that we're looking at. And okay, so this was the first part, was the 4th week. So then when the neurons are immature. Now what happens when the neurons are mature? Interestingly, the phenotypes flip. Okay, the neurons were hyperexcitable. They become hypoexcitable. Also, they become. So I'll show this. So we can see, this is now the control. So the control over the time developed. Now it's more excitable than it was at 4 weeks. And then the untreated, we can see they're less excitable. They produce less action potentials. And here I showed the minocycline example. We can see with minocycline, this flips the phenotype again, so it's much more like the control. We can see here in the recordings, control, untreated, and minocycline. And looking at the synaptic activity, again, we have this reduction of synaptic activity, so the neurons are less connected in the patient compared to the control. These are, by the way, 5 controls, and usually we have a direct family member. And with the minocycline, which reverses the phenotype, frequency of the events, we can see becomes more similar to the control, and also the amplitude. And now, again, this is a large drug screen, so these are the drugs that we have tested. We can see many of these drugs alleviate some or partially the phenotypes. So we can see with the excitability, again, the minocycline example, the control, the untreated, the minocycline, this is the excitability graphs. And looking at the synaptic graphs, how these drugs rescue synaptic activity. So we can see, this is an example with a CBD, cannabinoid oils, which are part of the extracted from marijuana. And then calcium imaging. Again, we have many features that we're looking at. Here also, the network burst frequency is lower for the untreated patient. And some of these drugs rescue this phenotype. And we have all these features that we're looking at. And we also, I didn't show before the network. So remember the networks, we're looking at, we're doing correlations between the neurons. We're actually using a method that is called time delay stability, which measures it looks for correlations between different neurons in the network. And then it looks if the time delay between them stays constant. And if it stays constant, it means that the neurons are connected. And we can give in the network here, we can give we can draw a line, which means the neurons are connected, they're synchronized. And so we have all these features that we have from the network metrics, for example, the time delay stability or the synchrony between the neurons. We can see actually the network is overly synchronized, but some of these drugs rescue this. Or we can see modularity, so how much is the network more like this, a modular network compared to this. And so we have many of these features. Actually, we have for each drug, we measure approximately 50 features, okay? So, and then we're saying, okay, so now we want to understand which of these drugs really helped, because we have so many features, how do we know, how the overall picture looks like? So we're using a principal component analysis, which takes a hyperplane, a 50-dimensional hyperplane, and projects it onto a 2-dimensional plane where our brain can understand what is going on. And there we can see, this is the results from the whole cell patch clamp. We can see the untreated patient neurons compared to the control. And we can see, some of these drugs that make the neurons much more like the control neurons. So if we look here, and I will also have a slide with summary, we can see, for example, folinic acid, CBD oils, methylphenidate, which is Ritalin, and others, okay? And we also have some combination treatments that we're still doing, but for example, CBD plus folinic acid and so forth. We're now, this is in process, we're still analyzing, but we are also looking, so this one was just the features of the patch clamp results, so this is approximately 13 features, okay? And now when we add the calcium imaging features, we can see it changes a bit the picture, but still, again, this is still under analysis. We don't have yet, the recordings are done, but the analysis takes some time. So we don't have all these dots also here. And eventually, we're also doing the network analysis, the network connectivities, the synchrony, modularity, and so forth. And you can see also, again, it changes a bit how the drugs look in this two-dimensional plane. But if we want to, this is right now summary with a patch clamp, because we're still analyzing the rest, but we can say that for this patient, the inversion mutation, CBD, folinic acid, the combination of CBD plus folinic acid, methylphenidate, and PTM all have a very good rescue. And then secondary, we can see amphetamines, aminocycline, soticlestat, glycerin, losartan, and potassium, magnesium threonate, and pregnenolone sulfate. So we have, this is another mutation. I will not go, I will just briefly show the graphs. I will not go deeply into the graphs, but we did this, as I showed, for these 5 patients. We're recording at the earlier time point, 4th week. We see all the patients have this kind of surprising hyperexcitability phenotype when the neurons are young. And as they mature, they become less excitable and less synaptically connected. So for this one, this is an example we see the hypoexcitability, which is rescued by CBD plus minocycline. You can see here the graphs. And we can see, for example, the excitatory postsynaptic currents, how connected that is, synaptic connectivity. We can see the example graphs. And this is, the entire drug screen. And again, we have approximately 50 features, approximately 15 from each of the, so 15 from patch clamp, 15 from calcium imaging, and 15 from network analysis. And you can see the calcium imaging. You can see here how the bursts look like. This is control, this is untreated, and this is elsewhere treated. And overall, again, the basic network metrics. And finally, we have these PCA that give us a suggestion of which of the drugs make the untreated neurons more like the control neurons. So we can see here, the drugs that worked. I can say, that there are some of the drugs worked for all the patients. Some are more specific to a specific patient, a specific mutation. So this is the patch clamp fully analyzed, calcium imaging, and the network still in analysis. And the treatments with the best rescue for this, this is the missense mutation. So we can see the soticlestat, magnesium threonate. This is lion's mane, it's just it's the mushroom. Losartan, potassium, CBD plus minocycline, and the second line is minocycline, elcerin, and NAP. And then just the summary of what we see with the others. So this is the 3rd patient, okay? So each of these dots again represents thousands of neurons recorded, or hundreds of neurons recorded. And we can see for this patient specifically, the best rescues are elcerin, NAP, PTM, CBD, and then CBD plus. So importantly, terpenese oils, we were able to only get a very small amount. It's very, very hard to get them into the lab. And we treated them, we had enough to treat only 2 of the patients. And they actually, for this patient, they gave some rescue. For the other patient, they gave even a better rescue than CBD alone. So, and this is another patient. This is the spice variant. And we can see, here the CBD plus terpenese are the closest to the control. Terpenes oils, by the way, are also a derivative of marijuana. They also exist in other plants, but they're highly concentrated in marijuana. It's in California. I think you can purchase them. In other places, it's more difficult, but it can be using, like with a clinician, can be purchased. So with this one, we can see the CBD plus terpenes, L-serine, D-serine, and then CBD, minocycline, CBD plus L-serine, CBD plus minocycline, and CBD plus THC. This is the active component of marijuana. Okay, so, and then the last patient, we can see also the graph, and best rescues are, CBD, CBD plus minocycline, Neuronal, sulfite, monocycline, and L-serine and CBD plus L-serine. So importantly, we do see drugs that repeat themselves. We wanted to use another model. We have the GRIN to be knockout rats, and we have done some behavioral experiments. So basically, we're looking at social preference, an object versus a rat. So the wild type rat would prefer to go to the other rat. And the social novelty preference, where you have a familiar rat versus a novel rat, and the wild type rat prefers to go to the novel rat. And we have an automated tracking and analysis system. And I can show briefly the videos. So we see that the wild type rat usually goes to the, this is the, usually goes to the novel rat. What was interesting was that the knockout rat actually likes to stay in the middle for a really long time. It takes him about 1 minute to start to walk over. And this is something that repeats itself. And we can see here, so this was the most significant phenotype. So we have like the knockout spends more time than the wild type with no stimulus, just standing in the middle. And then since minocycline was one of the surprises and also worked for all the patients, we started to treat, sorry. Oops. Trying to get the video to work. Okay. So we started to treat with the minocycline. So Ori, my student, is doing these experiments. He's through the water, is giving minocycline. And what we see is that, after the treatment, the rat behaves more like the control rat. And we can see the final graphs where we have the social preference test. We have the knockout, which just stands in the middle. We have the wild type, and then we have the knockout treated, which is more like the wild type. And then the social recognition test, where we have the familiar animal versus a new animal, and the wild type likes to go to the new animal. The knockout has no preference or almost no preference. And once it's treated with minocycline, he's again closer to the control, the wild type. And we're now continuing with other drugs. CBD, we had some problem to get to for in amounts for the rats, and I think we finally got them. So we're continuing with that, and we will do a few of the drugs that work the best. RNA sequencing. So we have done a lot of RNA sequencing, and I will show some of the results. Some of the results are still being analyzed. We have treated, with the best treatment options that at that time we had, because it was sent a few months ago. Since then, we started to treat with folinic acid, which we saw some very good results, but we don't have RNA sequencing, and soticlestat, for example. So overall, what we're doing is we have again the 2 time points, 4 or 5 weeks, and then 9, 10 weeks. And we're looking at the patients compared to the controls, and also the treated patients, which we haven't analyzed yet. But this, for example, is one of the patients, a nonsense mutation, sorry. And when we compare to the controls, we can see some of the pathway analysis. So we can see, for example, many pathways that are related to the synapse, which makes sense because we see that the synapses are affected through electrophysiology. And so we see, for example, synaptic vesicle cycle, vesicle cycling recycling, vesicle endocytosis, and so forth, and other pathways. Some of them we're trying to figure out how they participate. Motor proteins, some genes that are related to neurodegeneration, and so forth. And we have this for currently for 4 patients. We're doing some more. So this is the missense mutation. So for example, we can see nucleosomal DNA binding, ribosomal genes, nucleosome binding. Many extracellular matrix-related pathways appear in all the patients, okay? And then we have the inversion mutation. So here too we have extracellular and focal adhesion-related pathways. We have synaptic pathways, similar to what we see in the electrophysiology, and some pathways that are related to muscle, and others. And then we have the splice variant. So again, we see the ECM, many ECM extracellular matrix related pathways. Others, collagen, these are all a respiratory chain complex, but many pathways related to the extracellular matrix. And finally, we try to compare when we take all the patients together, compare to the controls, and to see, so here there were not many pathways. There were some genes, where there are approximately 30 genes that are dysregulated, that are together, in all these patients. But if we look at the pathways that are shared, most of them are, or all of them are related to pain or sensory perception of temperature stimulus, response to pain, sensory perception, and so forth. So these are pathways that are shared for all the patients. And at 9 weeks, again, this is the same patients, but when the neurons are more mature. So we can see this is the 6657 nonsense mutation, growth factor binding, calcium channel activity, SMAD binding, things that we're still working on to understand how they participate. This is the missense mutation, focal adhesion, which is related to the, how the neurons attach to the matrix, response to insulin stimulus, and more. And this is the inversion mutation, so RNA splicing, response to peptide hormone. We can see many of the pathways are affected. Again, ECM seems to be repeating, and also synaptic pathways. And the splice variant, we can see, again, electron transfer activity, the risk complex, many pathways are affected, approximately 1,000 genes that are dysregulated. And when we pull them together, so here, not many pathways are shared, but we can see many pathways that are shared that are related to muscle. So we can see muscle myosin complex, contract muscle fiber, and others. And finally, we did we did a high depth sequencing. So we're able to run to some of these. One of the patients has splice variant mutation, but also the other patient we're trying now to. We analyzed this. I got 2 days ago from one of the postdocs. We're analyzing now the isoforms or the variants that are maybe changed. So for example, these are the controls. There is one exon that is highly expressed in the controls. But if we look at the patient, this is a splice variant patient, we can see, for example, that one of the exons that doesn't get expressed in the other lines is highly expressed there, much more than the regular exon that is being expressed. We're now figuring out where is this exon, what part of the protein is it coding, and so forth. Okay, and with this, I will say thank you all for listening. These are the students that participated in the work. Our collaborators, so Dr. Tali Shkolnik from Rambam, who worked with us with the drugs, the list of drugs, to build the list of drugs that we are using for this. Our collaborators, Avi Schroeder and Shai Berlin in Technion, that we're working together as a group with a research network at To Be Cured. So this project was mainly funded by To Be Cured. Other fundings from the lab, and this is the lab, and thank you all for listening.